Third and final part of the blood notes, part 3. We're going to go back and reiterate this about the red blood cell cycle. So the kidneys on the left, the little diagram of the kidneys there, it says low oxygen tension at the proximal tubule. That's at the nephron, one of the millions of nephrons that make up the kidney, actually filters the blood. It actually detects low oxygen. So the kidney, basically for you, kidney detects low oxygen. In response, it produces something called erythropoietin, which is a hormone which causes erythropoiesis in the bone marrow. The red bone marrow then is stimulated to make more red blood cells, erythrocytes. So stem cells, the precursor to all cells, get turned into erythrocytes. So erythrocytes are made, your hematocrit goes up, that is, the more volume of erythrocytes in the blood goes up, and it gathers more oxygen. As those red blood cells grab more oxygen, it goes back and tells the kidneys, hey, we have enough oxygen now, so the kidney shuts off its erythropoietin production. That is negative feedback, like back in August. All right, we're not going to leave you hanging there. You can see that blood is totally recycled. The globin, the protein part of the hemoglobin, gets broken down to its amino acids and reused for all sorts of different things. The iron of the heme is mostly reused. You still need iron in your diet. And the biliverdin becomes bilirubin, and the liver turns bilirubin into bile. Remember, if you got liver damage, then you get yellow jaundice. So they can see the yellowing is because the bilirubin build up in the skin. And then again, the bilirubin becomes bile. Bile is used in the small intestines to emulsify fats that we digest. And then that causes your urine to be, or your, not your urine, your stool to be brown. Okay, the globin broken down, heme is converted, and the bile is what gives the stool its brown color. All that stuff I just said, here it is in writing. So again, red blood cells broken down. The spleen contains macrophages, which are constantly destroying worn out red blood cells every day. So this process goes on and on. Red blood cells only live about 120 days on the average. When heme groups unfold, the iron atom leaks out, producing one biliverdin molecule plus one iron atom for each heme group, and the biliverdin becomes bilirubin, which is used in the liver to produce bile, which causes feces to turn brown. Okay, we already said that a couple times, but here it is again. Iron is recycled in the body and not necessarily part of the bile, but you still need iron every day. So here's my question to you. What happens physiologically to an athlete who is training for a marathon and decides it's best to train up in Colorado Springs at the training camp up there? What's going on in her body? She's running, and if you remember the last slide, her kidneys are doing what? They're detecting low oxygen. So there's 1 point. Kidneys detect the low oxygen. Actually, it's 2 points. Low oxygen detected by the kidneys. Kidneys, in response, produce EPO, erythro, red blood cell, poietin, which kicks on erythropoiesis in the bone marrow, which means the bone marrow makes more erythrocytes. Boy, we've hammered this a lot. Preventing blood loss. So, like I said in class and demonstrated, assuming you guys were blood vessels, blood cells, when you get cut, the smooth muscles constrict around that blood vessel to make it smaller. Your platelets then stick to the collagen fibers in the connective tissue that's been cut, so they form a little plug. And the fibrinogen, which is throughout your body, which is inactive, remember, inactive because it's ogen, it becomes activated through cascade events to make fibrin, which is like a Spider-Man net, and it catches everything. So the blood clots then seals your little cuts and scrapes and your little abrasions. And like I said, the red blood cells, which get trapped in there, pile up, dry up, and become the scab. And the little yellow stuff which oozes out is now called serum because the clotting factors are taken out of it. It's plasma. Plasma is the fluid portion of your blood, but once the clotting factors are taken out of it, now it's called serum, and it contains antibodies and other hormones and stuff. The platelets do release a vasoconstricting chemical in response to damage of the blood vessels. What that means is when the blood vessels cut and platelets start sticking to the collagen fibers, they restrict a chemical which causes your blood vessels, vaso, to constrict and make it a smaller tube, so it's harder for you to bleed out. All right, platelet plug formation. Again, like I said earlier, platelets stick to exposed collagen fibers, which are in the connective tissue. Remember, your blood vessels are lined with simple squamous epithelial tissue, which is smooth and doesn't have the collagen there. When it gets cut, the surrounding connective tissue has collagen, and that collagen then sticks to the platelets, or platelets stick to it, and they activate this phospholipase mediated signal, makes them stick together, and then this inactive enzyme called prothrombinase, prothrombinase is an enzyme. It converts prothrombin, a precursor to thrombin. So prothrombinase is activated now. This enzyme is also activated in the cut, and prothrombinase turns prothrombin to thrombin. Thrombin then clips off the edges of fibrinogen, makes them sticky, so now it's fibrin. And this stays localized at the cut. All this cascade, this is a super short cascade of events. In the background, you see a little bit more details, which you see in college. But all this stays at the local event. It does not happen further down the blood vessels, otherwise you clot up and die. There's other checks and balances in the body which say, no, you're not going to release prothrombinase anywhere else but here at the cut. Again, the fibrin forms the mesh network and traps the blood cells. So once again, let's get this straight. Super simple, super simplified. You get an injury, you get cut. Clotting factors, we'll call them, release prothrombinase. Prothrombinase is an enzyme, ASE is an enzyme, which breaks prothrombin into thrombin. Thrombin then clips off the edge of fibrinogen and makes it fibrin, makes it sticky. Pretty simple, but really complicated in reality. But your body's amazing, so it does all this. How do we control clotting? We have anticoagulants in the blood which counteract this domino effect of the fibrinogen turning into fibrin and clotting your whole body up. We have clot retraction. Platelets actually have actin and myosin. Where else have you seen the two proteins actin and myosin? Right, in muscles. So the platelets will actually stick to each other and then kind of squeeze a little bit together. And then new connective tissue and epithelial is formed after the clot to heal it. And that's why you get scraped and 2 weeks later, you don't even notice it. Clot dissolution. How do you break up the clot? On the inside, we've got the stuff called plasminogen. Again, ogen, it's an inactive precursor to plasmin. It is converted to plasmin, and plasmin slowly breaks down the fibrin from the inside out. That's why all of a sudden your scab just kind of falls off. And you can get new epithelial tissue produced underneath that. Some other blood thinners, which help people who might have risk for stroke or heart attack, include aspirin, Coumadin, also known as warfarin, and heparin. So heparin is produced in the mouths of leeches, and it's an anticoagulant. A lot of times they'll stick that in a port to keep a needle or something that's inside of the body for a while to keep that from clotting up. You've heard adults being told to take aspirin a day to help thin the blood out so it doesn't clot. So Coumadin is used to treat blood clots, as in deep vein thrombosis. So your deeper veins, if you sit for a long time, like on an airplane or drive a lot like a trucker or something like that, you're putting a lot of pressure. You're pinching your veins, your deep veins, not superficial, but deeper. You're pinching them, and the blood might start clotting in there. If the red blood cells clot and stick together, then they can go to the lungs, causing a pulmonary embolus. An embolism is a clot which travels. So Coumadin is used to break those clots up and prevent new clots from forming in your body. So some older folks, chances of throwing clots because of maybe atrial fibrillation, they're on Coumadin. All right. Basic little diagram, again, showing you get cut, Your prothrombinase turns prothrombin to thrombin, thrombin turns fibrinogen into fibrin, boom, clot. Let's do a little label quiz. Do this on your own. A, right, erythrocytes. B, multi-lobed nucleus. It's purple. It's got to be the neutrophil, the most abundant white blood cell. C, ooh, C's kind of reddish. It's got 2 lobes. That must be the highly evasive eosinophil. It is. E, those little tiny dots, but very powerful in clotting. Right, thrombocytes. Oh, hey, there's D down there. It's a lymphocyte. It's kind of hidden. It got cut off a little bit. And E is a thrombocyte. All right, go to the following website, practice your blood types, study, and prepare for the test.